We are going to teach you how to make Captain America's Super Soldier Serum. Seriously, we've got a professor from CUNY and he has figured out the recipe. Up next on Science Goes to the Movies. Welcome to Science Goes to the Movies, a look at the stories of science and how they change our culture. I'm Lisa Bethkovitz. In the Marvel Universe, Dr. Abraham Erskine of the Strategic Scientific Reserve used his Super Soldier Serum to turn Steve Rogers from a 97-pound weakling into superhero Captain America. In our universe, Professor Sebastian Alvarado of the City University of New York wrote an excellent book called The Science of Marvel, and we've got him to come onto the show to explain how we too can make Super Soldier Serum. Maybe. Welcome to the show.
Thank you for having me. It's a pleasure.
So, Professor Alvarado, is Captain America like super strength possible? Please say yes.
I'd like to. If it was, I wouldn't be in the shape that I am in right now. But I do think there have been aspects of his biology that we've been able to create in the lab, let's say, specifically working with model systems like a mouse or a rat or something like that. So in some capacities, we can manipulate small parts of biology or physiology. And if we focus on those parts and we can kind of engineer them into a super soldier serum, yes, at least that's the best yes that I can give you right now.
But the mice are going to become super soldiers first.
We'll have our super mice more than anything else. And it's just so difficult to work with humans. So it probably won't be a very long time before we actually take that route.
So, the Captain America Super Soldier Serum hacks into human biology. And in our guest's book, The Science of Marvel, he writes that this can be done by forcing our DNA, and I get what that is, to send a different message to the RNA, a little bit shaky on RNA, but let's press on, so that the cells change the way they function via proteins. And now I'm totally lost, because I think proteins are something that's in my lunch, like tuna or burgers. So
a lot of this kind of is based off our fundamental understanding of how genes work. Genes and DNA and all that information exists within the cells of our bodies, specifically within the nucleus of any given cell. That information is static and it is unchanging. In fact, whether you have a cell taken from your eye, your cheek, or your muscles, they all have the same DNA, but that information is used very differently. And the way that information is used differently is how that static information gets changed into information in motion, let's say. And that's usually done through the intermediate of RNA. RNA, which is ribonucleic acid, is kind of this transient carrier of the information from DNA that allows that information to leave the nucleus of the cell. Then it encounters machines within the cell that can take that information as RNA and turn it into a specific protein. And it's that protein itself that actually has some sort of function. For example, you have a gene for insulin. It transcribes an insulin RNA molecule that's able to leave the nucleus. It finds machines within the cell that allow it to turn that message into a specific protein, which is the insulin hormone, which would then have some sort of function in your pancreas, or at least secreted from your pancreas and then in your body. So a lot of genes that code for proteins go through this flow of information. We call this flow of information the dogma of molecular biology because it's the one thing that we've been following for the last 50 years or so that happens to be consistently right. Does this mean all genes code for proteins? Not necessarily, but a lot of the ones that we have a good time understanding, in fact, do.
So you think that Abraham Erskine's on the genetic part of the Super Soldier Serum because it happens in a lot of different ways. He kept Steve Rogers' DNA the same, but changed the proteins that would operate in the cell?
I wouldn't say that much. I think, I got
guessing, understanding that we're talking,
right, of
we're
course.
Talking science about fiction.
We're not going to do this right now when we get back home.
I'd
like to, but maybe I will, who knows. But the idea is that I think, so really it was left up to for me to invent how this could have possibly worked. For me, it seemed to make most sense to actually modify the genes themselves. So in modifying the genes, you could change that message and then in turn change the proteins and the kind of function that they had. So I think, at least the way I kind of made it up in the book, we wanted to modify the actual genes and how that genetic information would then turn into protein down the road. Because if you modify the protein has a half-life. Eventually it'll degrade and be lost. Once it's lost, you kind of lose that, the effect. So I think more than anything, if you modify the genes first, they will carry that information long-term, as long as the cells that have been modified keep on resupplying themselves and dividing.
In real life, can we change our basic DNA?
So we change it all the time when we go out into the sun. Whenever we get a tan or anything like that, UV radiation can cause little mutations in your DNA, which is why you should always be careful, put on your sunscreen. So all kinds of things are constantly attacking your genome. The good news is that you actually have a lot of fail-safes and machines in your cells that help fix it whenever it happens. When I say machines, I think about enzymes, things like, not actual like little nano machines or anything like that. But you have these molecular machines that are responsible for fixing it. You're usually subject to a certain amount of stress from your environment, but you're very good at rebuffing it,
But we generally don't rebuild in a better fashion.
I wouldn't say so, at least in my understanding, no.
Okay. If you were, let's say, a mad scientist, Professor Alvarado, and you were going to create Super Soldier Serum specifically to change the musculature of Steve Rogers, specifically what genes would you be targeting?
So a lot of things that we can look at are actually just go into the literature and see what kind of research has already been done in studying muscles and muscle physiology. I would probably break or inhibit the genes that are specifically responsible for muscle growth inhibition. I would turn them off, and genes that increase muscle growth, I would turn them on. When it comes to genes that should be turned off because they inhibit muscle growth, things like somatostatin or myostatin, I believe there is a breed of bull called the Belgian Blue bull that actually has this very strong physique and a lot of muscles just because it has mutations within one of these genes.
What if you were trying to increase Steve Rogers' stamina? What specific genes in us create better stamina?
So for stamina, what I would mainly think about are ways of increasing probably oxygen carrying load in his blood, so allowing him to respire more and therefore have more energy to produce the energy that he needs to sprint, to jump, to fight bad guys.
So endurance is all about oxygen?
Well, I would say a big amount of it is the ability to traffic oxygen from the lungs to the extremities of the body, so that way those muscles can function from respiration. The more respiration you can do, the more energy you can provide to the rest of your body. One way of doing this is by increasing red blood cell count in your blood, which would increase the overall surface area that's able to carry oxygen and therefore increase stamina as well as endurance.
So can a single protein change what I am?
It depends. It depends how important that protein is, where it's needed, how it's needed. I mean, if you're thinking of a protein that you need to go from a 2-cell embryo to a 4-cell embryo, yeah, it's really important because if you can't get past that 4-cell embryo, well, you're never going to make it to 37 trillion. So for that, it's obviously really, really important. However, I would say, there are a lot of compensatory mechanisms at play. That if you break one gene, there are ways that your own biology fixes itself. In some cases, specifically rare cases, you have things like genetic diseases that can be heritable within a family, and they often have some sort of, they have a way of changing the pathology of an entire disease, and sometimes that can be resolved down to a single gene being mutated or not mutated. So yes, in some cases, no in others. Surprisingly, despite having some 20,000 genes in your genome, that really only makes up about, let's say, 20% of your genome. So there's a lot of unknown. There's a big question mark hanging over the rest of it. There are a lot of people understanding it now. We're trying to figure it out. But like I said, we understand that, gene, RNA, protein a lot better than we understand that other, let's say 80%. And a mutation in any of those genes, I don't know what would happen. That's really the answer that I've got. I don't know. Chances are a lot of nothing might happen, but who knows? We might also get a Captain America.
But tangential to this for a screenwriter, if you were a screenwriter, and I know that you sometimes advise screenwriters, so generally, if you're going to do a fiction, a temporary change is the change to a protein, and a heritable and consistent change would be a change to the DNA.
Yes, to a certain extent, yes. But I love saying this, it's more complicated than that. Because if that cell is only going to last 2 or 3 divisions and then disappear, then it's now temporary, right? If that cell is, let's say, part of your stem cell pool that's regenerating, it's going to stay with you till you die, then yeah, it's definitely a lot more permanent. It all depends on the type of cell, where that cell is, and how active it is in dividing. But yeah, if you definitely want to make a much more, let's say, change to the foundation.
Of your character, of talking fictional.
Yeah. No, I'm thinking about the real thing.
I know you are.
If you want to change something more foundational at the molecular level, DNA is definitely where you start. And that DNA hopefully is in a cell that's very stable across the lifespan of the individual.
So how do you jack into a cell?
Yeah, so there's a lot of ways. Ways that we use in our lab, and other labs as well use them. This is something that's changed a lot in the last few decades. For starters, at least what I was kind of growing up with when I was in graduate school, is you would have these protein complexes that bound to DNA. And then you would have one specific protein that can only recognize, a few letters of the DNA code. And then that would hug onto that piece of DNA. And then you'd have a different protein that would only recognize another few letters. So you would almost make these little train carts of these proteins so that way you could recognize a large span of DNA. That was a lot of work. And then you would attach those train carts, those molecular train carts to a molecular pair of scissors to actually destroy the DNA or to break that gene. Like I said, that's a lot of work. Things have changed rather rapidly in the last, 5 or 6 years, specifically with this genome editing technique called CRISPR, which I'm sure a lot of people have heard about, because it's kind of replaced that, very complicated train track that train cart that binds to many parts of the DNA to just one protein that binds to one RNA molecule. And that together is enough for it to have surgical precision to target almost any gene it needs to in any genome. It works really, really well, and not just in model systems that we've been using for decades, not like just mouse, fruit fly, worms. These are things that they're very good at because we've been working with these animals forever, but they work really well in butterflies, cichlids, you name it. So it's very, very robust tool set that's allowed us to do a lot of really cool biology.
CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats.
I'm glad you said it, so I didn't have to. Yes, it does, yes.
Does that describe what it is, or does that describe what it does?
I think it's actually a bit of a description of where they were originally derived from, where they were discovered. There are a lot of people that have been involved with its discovery and its engineering. The clustered repeats are actually in these spacer sequences of DNA that bacteria use. So this is originally derived from bacteria. And what they do is they use these spaces between these palindromic repeats to store genetic information of viruses that might be infecting those bacteria. So when they did, they were able to use that spacer and integrate it into a CRISPR RNA, which would then associate with the CRISPR-associated protein. So all of this would work together to then attack that virus, so that way it wouldn't be able to infect the bacteria. In much simpler terms, it's really just a protein that binds to an RNA molecule, and the two of them work together, and that RNA molecule has complementarity to sequence on the genome that it is targeting.
So you were talking about, when you were talking about molecular scissors, you were talking about a mechanical process.
Yes.
Is CRISPR a chemical process?
It's just like a lot of other proteins. It's just it's a protein that has enzymatic activity. And it is like a pair of scissors. It's a molecular pair of scissors. So wherever you take that CRISPR-associated protein, it's called, we usually call it Cas9, wherever it finds itself guided, it will then cut the DNA that it gets close by. So the RNA molecule is kind of like it's information that allows you to let it seek out the right gene. But once it gets there and it binds that DNA, it cuts it up into pieces.
And when you say let it, does it automatically seek out the gene rather than you having to physically find it?
Yeah, so with these other approaches, you have to imagine, these train carts of proteins, right? That's a really big thing, right?
Yeah.
And you either have to trick the cell into producing it itself, or what you have to do is traffic that large protein past the cell membrane, past the nuclear membrane, because it has to get to the DNA, and into the nucleus of the cell, and then search through 23 pairs of chromosomes, some 20 gigabases of DNA, and then it finds it.
I can't even find scissors in my house.
There you go. So it's a good thing we have these proteins that do it for us. So it's very, very difficult. But with Cas9, it's small, it's compact, it's very robust. Those other approaches, they often miss. They might not be very good, and a lot of work gets invested in them for them not to work. With Cas9, it works rather well the first time, and if it doesn't work the first time, it'll work the second time. Do you know what I mean? And there's not a lot of effort and labor that goes into that. So it really did revolutionize our ability to just manipulate genomes in general.
In the Super Soldier Serum, they use an artificial element in it. Can we put artificial elements into our genes? Can we accept a synthetic protein?
So proteins themselves are likely to degrade over time. We can introduce new genes that aren't normally produced by us. You can modify old genes. There are strategies to do things like that we can do in the lab right now. But I wouldn't say that there's anything again, these proteins are more transient than synthetic. They need to have the information the blueprints to create it in the cell itself. So I don't know if you could really incorporate synthetic proteins. The body in general has a good way of cleaning itself out whenever something like that happens, or biology in general has a good way of cleaning itself out, or dying. So.
So, but these genes that were changed with the Super Soldier Serum, if Bruce Bannon or Steve Rogers have children, are they passing these genes on?
In the comic books, they do, because it's exciting. Everyone wants to know what son of Hulk or son of Steve Rogers is. So really, what you're expecting in that scenario is that, in addition to manipulating all of these genes in muscle, in the brain, in the bone marrow, all of those things, you also want to be manipulating the genes in the testes. So you're really going after, the family jewels, quite literally. So those mutations and the way that they seem to be administered seems to be very locally changed, at least in Stephen Rogers' case. So I don't know, which isn't to say maybe it is, maybe it's not. I know we're in fiction, but who knows, really. There are some really neat examples out in the literature today that looks at transgenerational inheritance independent of the way genes are actually changing. There's some work that was done at Duke that looked at how the fears that are associated with specific experiences in fathers can be passed down to the next of kin through the sperm, through other chemical means of modifying the DNA. And that's really fascinating work, because it really means what you're doing could very well be affecting the next of kin without really knowing how and what. So maybe being Captain America is programming Stephen Rogers' testes to give birth to other super soldiers. Words I never really thought I'd be saying. But,
on TV.
But yeah, on TV nonetheless.
But it's interesting though what you say, because these genetic changes, say I have a genetic change, let's say to my shoulder, it would have to work into it would have to have enough time to get into my reproductive organs before it would become a genetic trait that I passed on.
Which sounds like science
dyeing
fiction,
my hair
right?
will never anyway.
Well, you don't know. I mean, it sounds like science fiction, right? But take this one example. I think it was done by Brian Diaz and Kerry Ressler. They found that they associated the memory of they associated the memory of a foot shock with a specific odorant, and this turned that gene off or turned it on in the olfactory bulb of a mouse. That experience was able to travel from that part in the brain over time and space to their testes so that they could sire young who were then afraid of the same odorant. So they would freeze up when they smelled that same smell. So it's almost like how it happened, right? It's science. It's in front of us. The data's there. But it's so hard to believe. And that's just the science. We're not even talking about the fiction. So, you know, if we're talking fiction, yes, of course, let's go there. But I mean, it's just really fascinating to even understand it in real life because apparently it's happening. So maybe it could. But based off the genes and how they're being modified, and the process that's involved with that modification, it's hard for me to draw the link between what's happening here in the testes, even if it's happening in nature, in the way we look at it today. So,
But it's all happening to mice, so is the future all Pinky in the Brain?
I mean, well, while we're still working with mice, we can explain all of these things in mice, I guess. And we have a lot of good reasons to continue studying them, but we also study other model systems that have the ability to pass on these things across generations. There are species of invertebrates, I think C. elegans, another worm that's able to pass things on transgenerationally. It's all very exciting work.
It is. It is. And in the first Captain America movie, Steve Rogers is found frozen in the Arctic. So let's talk cryostasis. First off, what is cryostasis?
So my understanding of cryostasis is that it involves the freezing and preservation of an individual organism, organ, tissue, or cell. And this for the ultimate purpose of having it extend its life or being, thawed out afterwards and still alive.
So things frozen but not dead.
Frozen but not dead.
So in The Science of Marvel, you write that Steve Rogers is a really sweet guy, and that matters for cryostasis. Why does sweet?
This was my tongue-in-cheek eye roll moment while I was writing that. I said, I'm going to put it in the book. I don't care what the editor says. I was doing so I actually was discussing how Steve Rogers was able to survive freezing because he adopted a specific way to preserve his tissues, the way that wood frogs actually survive freezing cold. This is actually work that I kind of borrowed from a colleague and collaborator, Ken Story in Ottawa, and he studies these hibernating vertebrates, specifically the wood frog. And in his work, he actually showed that one of the biggest stresses that cells undergo when they are freezing during bouts of hibernation and things like that is that they have to form ice in their tissues. And the formation of ice itself is the formation of a crystal structure. And the crystal structure forming in your tissues is like, it's literally a sharp object that's growing out of your own flesh, like these little tiny knives growing in your tissue. And that in itself creates a shearing force that destroys tissue. So freezing, sure, fine, but thawing and that freezing is basically creating crystals that are tearing up the tissue from the inside out. And this could really destroy something. So what frogs do is they actually pump, what these wood frogs do is they pump sugars from the inside of their cells to the intercellular space. And that actually increases the viscosity of the water and actually stops the formation of this, of crystal ice.
Just the way sugar freezes differently in your freezer.
Exactly. So the idea here is that you're basically stopping the formation of ice and therefore eliminating the shearing force and then stopping the destruction of tissue during freezing. So this is one strategy that, would work if Steve Rogers broke down all of the glucagon in his liver and was able to pump all the sugar out into his extracellular space. He'd be a much sweeter guy.
Or was just super nice to everybody.
He's just a nice guy.
If his veins ran sugar, which I understand they do.
It's like all you're missing now is that one scene in like Civil War where they're fighting and then he's like, ooh, he's like licking his thumb.
But so then if Bucky Barnes is captured by Hydra and frozen and thawed and frozen and thawed and frozen and thawed so many times to do assassinations, he would be under considerable more physical stress than being just the act of being frozen.
Yeah, definitely. Because you're basically, again, you're growing those miniature flesh knives in your gut every single time. I don't know if he's resisting it. Perhaps there's something special, kind of like this, pumping sugar out. He's not as sweet as Chris Evans and Stephen Rogers, but maybe he's pumping it out, or at the very least, he's recovering and he's
Yeah.
healing from it.
Yeah.
Which means that every time he thaws, he's going through a lot, or yeah, pretty much every time he thaws, he's recovering from a lot of micro injuries and tissue damage, which he can do because
Yeah.
he's the Winter Soldier and he has a metallic arm. You can do that in comics,
Right.
but I mean, it's obviously maybe why he's never smiling, and he's always in a bad mood.
Right, because his tissue has been sheared.
Yes.
So, all of our wonderful sci-fi intergalactic travel depends on cryostasis. So, Captain Alvarado, if your mission depended on cryostasis, how would you do it?
I would send somebody else. I wouldn't do it. To be honest, I'm not entirely convinced at how it's currently being done. I've heard a lot about, and I think I wrote about it in the book, about vitrification. So I guess supercooling it very, very fast, so that way you don't have the opportunity to form these crystals of ice. But then what, right?
No.
Then you're just an ice cube.
Right.
I would recommend against it and just enjoy life on Earth and try and preserve Earth a little bit longer.
Thank you so much for spending time with us. We are out of time and we didn't even get to talk about the Hulk. I hope you will come back
I'd
another
love
day and
to,
talk
yeah.
about the Hulk.
It was a lot of fun.